Hello. How is everyone doing today? Oh yeah, still dark. Okay.
Noisy in the background here.
Okay. All right. All right. So Sushmanth and Jyothi, welcome, and then Morgan, also welcome.
Yeah, it would have been really nice.
Morning.
Good morning. All right. Well, Sushmanth, I don't know if you have any final updates for GSoC. Today's the day everyone turns in their projects. And I talked to Himanshu on Thursday, so he's busy turning that in. So it looks pretty good, actually. He got done stages 1 and 2. So those will be up on GitHub soon. We'll be able to interact with those things. It looks like a social, like Sushmanth and Jyothi have been working on their on Hacktoberfest activities. So thank you.
Thanks, Bradley. Today is my GSoC submission actually.
Okay.
Last day. I just want to give you a heads up. Like I have pushed the whole code. I have written a blog about final report also. I have added. I hope you have seen the report. I'm sharing it in the group. The results are pretty good right now. I have shared a link. Could you see that?
Yes, I see it.
Yeah, I have added the result. I have actually. I will share my screen and show you.
Okay.
Is my screen shared?
Yes. Yeah, there it is.
Yeah, this is a complete model training. I worked on two type of extensions actually: box prompt, and point prompt. The results are like this. This is the original ground truth and original image ground truth, and its nucleus point. These are the results I was getting when I was giving a bounding box as a prompt. I was getting these results. Actually, SAM is segment anything model is trained on CNGS data from cell tracking challenge data set.
Okay.
I have added two methods. One is bounding box prompt and point prompt. This could be used for live fine tuning of a cell. I mean, we have a microscopy live imagery. If you want to analyze cell shape, etc., things, you can use this model. I have added the whole thing. I have added the like weights of it as a separate thing. I have added the step-by-step tutorial on Collab also. For everything, these are the implementations of notebooks. This is point from these are the things, Bradley. I have worked on the single GPU support and multi GPU support also. This is at starting stage only. This could be extended for next GSoC also. Jyothi have also worked on this thing. He was parallelly contributing to it. If you see in the contributors list, and DevoNet also have pushed some code. Let me push it. I have worked on it. Badly, I couldn't write the paper with all these things. But in coming next couple of weeks, I will work on that also.
Okay. Oh yeah, the papers. That always takes a while to kind of get. Yeah.
Yeah. Writing them all is like with vocabulary and stuff. It is so hard to me. I didn't mention it, but yeah. Yeah, but we have to shoot the DevoNet code also. The only problem is right now is the model hasn't been trained now. We need to train the model actually because of GPU issues. I couldn't train the model.
Okay.
My college, whichever college I'm studying right now, they are providing me with some GPUs actually next week. I will take those GPUs and I will train the model.
Okay.
This costs a lot, Bradley. These GPUs I have seen. For 1 month subscription, they are asking around $100 in Collab.
Oh, wow.
So I requested from my college.
Okay.
They are able to provide it,
Good.
but they don't like they are working on something, they will provide next week. I will train that model also, which I have proposed in my proposal. But right now, CellSAM is working right now, which is fine-tuned on the microscope image. It is not only meant for C. elegans, Bradley. It could be used for other cells also, plant cells, whatever I have trained it on the whole data set of cell tracking challenge data set has different type of data sets, Bradley, around like 13 model organisms out there. It is trained on them also, not only C. elegans. It will be able to segment them also in a particular and I think D. Mellinger, I don't know the exact pronunciation of D. Mellinger or something. It is that model organism. I have trained it on that also. It consists of around 2,500 cells because C. elegans has only 360 cell stages, but it will go up to 2,500. Our model is trained on that data also, so it could work very perfectly. And the SAM is trained on the color image. When you take a grayscale image, has only one channel and it is like grayscale images. So I have fine-tuned it and make working to it. The demos are over here, Bradley. In the repo CellSAM repo, I have added the all README file, how to train it on particular their own data set, and yeah, those are my updates. The code is ready, Bradley. Next week I will train the DevoNet model also, and I will show you the report.
Okay.
But you need to give the what we call evaluation for the final evaluation by next week.
Yeah, I'll take care of that.
That's okay.
Yeah.
But I just want to let you know I want to contribute more. Could I do it right? I mean, this is GSoC period is completing.
Oh, yeah.
Next couple of months also, I will attend the meetings and I want to contribute. And Jyothi was also interested in the next projects I have in mind. I will discuss through further because SAM is just we have tried the model, so we can extend it for next GSoCs or we can work it on personal papers because segment anything model provides a zero shot segmentation methods, and this could be used in the microscopy image analysis because protein structures etc. It can be used very perfectly. We can extend it up to there and we can use it. So if you are okay with it, I want to collaborate and work with you furthermore. Is that okay?
Oh yeah, that's great. That would be great. Yeah.
And the papers also, I will try to write as soon as possible, Brandon.
Yeah.
Due to this code, and I have learned so many things in this fine GSoC project because learning biology is a different kind of stuff, and integrating these two domains, deep learning and biology, is like something big I have thought. And it helped me a lot, and it is helping me to go for further studies in the computational biology. I was thinking to apply for masters in this subject and learn and dive more. And finally, thanks for providing the support for GSoC period. It could have never happened without you with the support actually.
Oh, yeah,
I have
yeah.
learned a lot of things. I want to maintain DevoLearn and work on different things also. I have worked on right now Hugging Face has been stopping every day, right? I have solved that problem also. I have posted it on the render. I will share with you.
Yeah.
Apparently, this is the link. I have posted it like my dev did, right? Exactly, I have posted in another dev services.
Okay.
This is free right now. If Hugging Space stops also, we can use this code. That will be fine. And DevoNet documentation also, I will provide you with that. Maybe if you are free, I will attend the next week Saturday meeting on the orthogonal lab meeting. Is that okay?
Yeah, that would be great to give a presentation on it. Yeah.
Yeah, I also want to give a presentation. Right now, I was fed up with other college works, so I will show you that also.
Okay.
This is, I will show you the demo. Right now, I don't have the demo, but the code and everything is fine. I will train the DevoNet model and also I will show you my next couple of weeks. And paper thing, we will discuss it further and we can make progress.
Okay. Sounds good.
If you see the CellSAM repo, you can find everything. This is the whole documentation of CellSAM.
Okay.
externally. What I have learned and how I have implemented on my dataset shared here. And this is CellSAM repo. This is CellSAM.
Yeah.
First one is the documentation, second one is the report with the complete code and weights files of that. And I will post the CellSAM in like Hugging Face spaces also, because we have right now DevoLearn has 3 models: cell membrane, unit segmentation, lineage, population. With that, CellSAM will be also added into Hugging Face spaces. I'll work on that. I have found a method to reduce the file size and post it on Hugging Face spaces.
Yeah.
So I will work on that very yeah, by next week, my work has been completed on CellSAM, right? So I will, next couple of weeks, I will try to write as a paper to it and post it in ourselves. Because this is not only important for you, but also important for me, because I want to apply for masters. I need some papers to work on this thing. So I will continually collaborate and I want to work on this thing. Is
Okay.
that okay, right? That's the whole thing.
All right.
Next week on orthogonals meeting, I will attend and I will show you the presentation of how to work, how to use this model, etc.
Okay. Yeah, that sounds great. Yeah. So that was a great update, Sushma. Thank you. Yeah. So let's go through some of those things. I'll share my screen. Okay. So I'll share my screen and we'll go through some of the things that Sushmant shared. So yeah, this is, you can think of GSoC as a stopping point. If you're interested in continuing to work on things, everyone who does GSoC is welcome to do that. In fact, it's encouraged because you create this thing, this artifact, and you put it out into the world. But if no one uses it or no one does anything with it ever again, it's not very useful. So and I know that Sushmanth is working on some papers associated with some of these tools that he's built. So yeah, let's work on those next, and work on some finishing touches on the projects and things like that. And Jyothi, welcome. I'm glad to see that you're contributing to Hacktoberfest. I saw your pull requests. Also, yeah, if you're welcome to to attend the meetings and work on things. And we'll be doing the next year's GSoC projects. We'll start talking about those in January. So it's always good to be ahead of the curve on that.
Sure, thank you.
Yeah. So we have a couple of things here. The first is the gist. This is Sushmanth's gist, which is like a GitHub repository for sort of submitting things to places. So this is going to be submitted to the GSoC project, the final project. So this just kind of talks about the different models. And this will be useful for people who want to use the models as a very high-level documentation. Again, the papers will be more detailed in terms of the details of the models, different attributes of the models, and going through every nut and bolt instead of the broad overview. So this kind of talks about CellSAM, the segment anything model, which is what CellSAM is based on, a little overview of the model architecture. Now for the paper, one of the papers, I guess we're working on the CellSAM paper, we would want to expand this section a little bit and have like some details about each part here, where it talks about the prompting mechanism, zero-shot generalization, and visual prompting extension. So those are all, and then the architecture here, which, I don't know, we can make a better figure, but I think going through each of these parts is good. And then, then there's this sort of aspect of using it in biology. So we have the harness a segment anything model for microscopy, fine-tuning SAM. So this is where SAM is getting trained, unveiling a new era of cellular analysis. This is about C. elegans, and then this shows like a step-by-step on how to work with the data, basically. And of course, we can, in the paper, we can maybe do some benchmarking here at this part, where we kind of show that not only how to use it, but like, how it compares with well, I don't know how we would benchmark it, but that's up to Sushmanth to make that determination. But,
Hello?
yeah, yeah.
I have worked on other things also, comparing with unit structure and DMAPNet. Those are other tool trained models. Our model is giving better performance now. I don't know how to create those graphs. I asked Hough, he told like he will help me with that on this coming Thursday.
Okay.
So I will work with that, and I have trained the model, but I need to make the loss graphs of both of those models. So I will make it, and I will host the comparisons also. Mike was little busy last week; he couldn't help me with that, but he told he will help me in this coming Thursday. So at that time,
okay. Yeah, that's great. Yeah, that's good. Yeah, the graphs are very specific to benchmarking. So, I mean, you can find them in any paper, but you have to actually plot the data. So, and I can, if you need any help from me, I can help you with that too. But so there's the segmentation data class, CellSAM model architecture, loss function. So this is all, and then you have the very general stuff at the top, the more specific stuff down here. And again, we'd want to expand some of this for the paper. This is really kind of like the the basis of the paper. It's just kind of the outline. So we need to flesh out each part. And then, future improvements, hosting, which of course has been an outstanding issue for us because we used to host things on what was it called? Whatever it was, and then that went down. That went away.
Hugging Face.
Well, we're hosting now on Hugging Face. Yeah. So I think Hugging Face will be around for a while. I think it's pretty well run and funded, and it's explicitly open source. So we'll have this hopefully for a long time. We'll be able to work on the models there. So this is good. And then thank you for the note of gratitude. Yeah. So these are just different sources in the project page, and that. So that's good. I like that. Let's see. This is the same one. This is on render, and so this is okay. So yeah, this is the sort of the technical preprint that we had before, and this had all right. Now this is hosted on render, so this kind of goes through this. Oh, so we can do this with the models. We can actually go in and open up a file. And okay, there's a default file, and you're able to index it in the interface. So I can find this sort of a point on this in this cell body, so I can get a quick assessment of each cell, centroids, other areas of the cell, the boundary of the cell. And it's in this coordinate system. I guess it goes up to y0 to y250 and then up to 350 on the x. So it's all like that. And then you can normalize it later. I mean, you ideally want to get the numbers, but you can do it this way. And that would be fine. This is nice. And then, of course, we can set the threshold, the different levels. So this is kind of, well, yeah, I mean, you'll see here. It'll come up and give you, kind of a different segmentation, I guess. And then the nucleus segmentation, which is a different type of segmentation where you're segmenting out nuclei. This is kind of where we have you can see the signal. It's doing what I would think it would do. These images that I think we have aren't specifically stained for nuclei. If you look here, the cell membrane segmentation, we don't really have a good stain for nuclei. Like sometimes people will put in a nucleus stain, and so something will get expressed in the nucleus and it will have a fluorescent signal. And so the fluorescent signal will be quite focused. And so you can actually segment by that if you have the images. Some data sets use nucleus staining, some use the membrane staining. So that's just a matter of getting the right data set, or you have the option to do that. And then predicting lineage populations. This is where we had and this is from previous years, but updated for this year. This is where we have the fluorescence microscopy for each cell. These look like actually more like nuclei stains, and we can actually track them in this way. So this is nice that each of these models are hosted like this, and then we can actually grab data from our computer, local computer, or look at the demo and see what it's supposed to look like. So that's great. So we have that, and then finally we have this. This is the CellSAM. Okay, this is the repo. So we have all of this on DevoLearn if people are interested in contributing. We have, of course, our Hacktoberfest project, which is this annotations, and then we have CellSAM. So CellSAM is where, and it looks like Jyothi and Sushmanth have been contributing to this. This is one of the models that Sushmanth was talking about, CellSAM. This tells you how to install it. And then, there's always this issue of training on GPUs. You have to pay a lot of times to get access to GPUs to train these models, so they don't just come out of thin air. So I'm looking forward to seeing that. That'd be great. I think we did this before with training the models. It's always depending on what kind of data you want to put in. It depends on the sort of the variety or the breadth of the model that you want to build. So if you want to train it just on C. elegans, you can do that, but we're trying to train it more broadly on different types of cells. And as you've seen in the meetings, our cell types are very wide-ranging that we have available. So we have all sorts of cells come in all shapes and sizes and different features. So it's not an easy thing to train them for. We're kind of a general purpose model. So that sounds great. Thank you, Sushmanth, for being a good GSoC participant. Thank you, Himanshu, for your GSoC project. And thank you, Jyothi, for joining us. And look forward to some good work in the future. So we'll work on the papers. That'll be good. And we'll get those out. And again, like I said, I think we have everything in hand. It's just a matter of writing things up and getting the technical details worked out. And so that's great.
Maybe from next couple of weeks, Jyothi will also start contributing to DevoNet, etc. And we will help with that. Those are my updates. Next week in Octogonal's meeting is there, right? It is on Saturday, right?
Yes, it's Saturdays at the same time as this meeting. Okay, it's at a different link.
Perfect.
Yeah, it's at a different link.
I have the meeting link. I will attend through that.
Okay.
Anyways, thanks Bradley for the whole support. I want to extend my gratitude to you particularly. Thanks for learning. I have learned a lot of stuff in this period, like learning new things without. And I just want to tell this thing: if we don't know anything, also that's okay. We can learn all these things and go. This has helped me a lot. By contributing to DevoLearn, I have learned a lot of things. Thanks for the continuous support. That's the whole thing I can say. Really thanks, Bradley. Those are my updates. I will update you in the next week meeting. Thank you.
Oh yeah, no problem. Thank you. All right. So that's great. That's our GSoC for this year. Wrapping it up. I think we did a lot of good work. We had, we worked on 2 projects. We worked on the DevoLearn, and I think we made a lot of progress on that. We also worked on the DevoGraph, and we made some progress on that. It's still kind of some things to be worked out there. It's a hard problem, really, and we're getting there, though. It looks like Himanshu has a lot of the embeddings that we've been thinking about. He's kind of worked those out and kind of figured out how to plot those out according to some of the methods they use in topological data analysis. So we'll continue to work on that as well. I'm looking forward to getting some of these papers out. And So, are there any other people want to mention anything or have any news or things that they want to talk about?
Oh, the good news is I'm almost finished my writing up the first draft of my candidacy project.
Okay, yeah.
And my structure for the epithelial tissue mostly works. Well, it has some issues, but at least it kind of stands on its own and I don't know, produces a stress strain curve or something. Very basic.
Oh, great. Yeah, yeah. Well, that's good that you were able to get somewhere with that.
Yeah, I was. It gave me a lot of hassles.
Oh, I'm sure, yeah. Morgan or Jesse, how are you guys? That's pretty early for Morgan.
Good, thank you. Yeah. Sorry, I'm making school lunches and things, but great to see all those projects and yeah, good to be here.
Oh, thanks. Yeah. Okay. Well, let's move on to some other things. I'll share my screen. So last week we talked about epithelial cells and some of the measures that people have made. And interestingly, no one's ever made a data set that's been like, size and shape parameters. We want to maybe do things like build a model or characterize the topology of some epithelial cell, set of epithelial cell layers or something like that. And I talked about some of the ways in which epithelial cells sort of, some of the formations that it makes in the human body. So, we had the long thin cells, we had the square cells, we had layers of those which are of different type. So you form all sorts of different types of epithelial tissue that do different things. So, sometimes you find them in the gut, in the intestine, stomach, on the surface of the skin, and they serve all different types of protective and other types of functions. So that was kind of interesting stuff. So this is kind of an interesting set of papers here. This is a paper that I found. It's from Physical Review Letters. It's kind of a biophysics take on epithelial cells and epithelial tissues. This is called Non-Affine Mechanics of Entangled Networks Inspired by Intermediate Filaments. And so this is a paper that came out recently. It was this summer. So when they say non-affine mechanics, that means that they're basically looking at what they call entangled networks, which means that they're not, straight arrays. They're kind of tangled together. You can see these kind of strings that they model here. In the way that these cells are oriented, they're oriented in different directions. They have these strings that are kind of, these cell outlines. They're just basically not like straight. They have a lot of, they're tangled up and have a lot of different dynamics like that. And so there are these intermediate filaments here that serve a role. The orientation of the epithelial cells here serve a role. So you can see the intermediate filaments coming out of the cells here between the cells. And so they're not, the array is not as straight as I showed you. Last week we had these monolayers and these layers, multiple layers of cells. And, it kind of looked like it was a nice, neat layer, but they're not, as it turns out. I think we talked about this with respect to mitochondria a while back, where mitochondria, the textbook definition of mitochondria, it's like you have a cell and you have a couple of mitochondria in the cell, and then they're kind of independent things. They're kind of like these little kidney beans in the cell, but actually they form a network, and the network is a lot more complex than what you see in the textbooks. So this is So the abstract reads, inspired by massive intermediate filament reorganization in superstretched epithelia. So this is where you get this filament reorganization. We examine computationally the principles controlling the mechanics of a set of entangled filaments. So these are these entangled filaments here, and they're in this layer of cells, whose ends slide on the cell boundary. So the ends of the filaments slide on the cell boundary, and that's part of the physics of the layer. We identify an entanglement metric and threshold beyond which random loose networks respond non-affinely and non-linearly to stretch by self-organizing into structurally optimal star-shaped configurations. So they have these star-shaped configurations which are optimal. It looks something like this, I think. So this kind of outline, they have this specific characteristic configuration that they basically stretch into. A simple model connecting cellular and filament strains link emergent mechanics to cell geometry, network topology, and filament mechanics. So they're interested in the topology of the network, which is the star shape, the cell geometry, which of course changes as we've talked about, and then the filament mechanics, which are separate and apart from that, but they're basically these tangled filaments, and so they have a different mechanics than you might expect from maybe like a straight array.
I've been reading something that says that when cells are compressed, around about 70%, like when they're, I have the reference, then the intermediate filaments come into play. They don't come into play. I have a reference for you. It's quite long, but they've investigated intermediate filaments, and they're attached to desmosomes
Okay.
on the sides of the cell, and they don't affect the mechanics until the cell is quite compressed, 70%. I don't know. I'll find the reference. Like I said, it's quite long. It's actually a thesis, but it's interesting. So it's vermentin, the intermediate filament vermentin, and perhaps another couple of kinds of intermediate filament.
All right.
Yeah, I don't know where I put that. I think it's Schaefer's. Anyway, I have it in my write-up.
Yeah, be interesting to see.
I'll send it to you.
Okay.
Or
yeah.
find it. Maybe I should find the reference and put it in the chat.
Okay. Yeah, that'd be good. Yeah, I've been meaning to send you those references on the epithelial stuff, but I've been busy, and so we'll have to get that together.
That's okay. I know about busy.
Okay. So yeah, so let's go back to the paper. So yeah, this sort of system is in a superstretch regime, so it's not like a normal function, but they show they use that to show kind of how this works. So epithelial tissues are cohesive cellular sheets lining free surfaces and multicellular eukaryotes. They're involved in crucial physiological processes such as morphogenesis, protection, secretion, and absorption. They serve as biological barriers, but also protective. So they have this protective function, but also they do other things. And then they talk about here microskeletal actin filaments are short, stiff both to stretch and bending, so less than 1 micron. So it's like pretty small, but they're pretty small in diameter, so they're kind of like a very, very, very tiny rope in some ways. And so they kind of attach between different cells. So then they talk about binding to a variety of specific crosslinkers, including myosin motors, and turnover within minutes. So they absorb and resorb. So you get new ones every so every few minutes. So these are, not only are they tangled, but they're regenerating constantly. Microtubulars are long, stiff, dynamic, and also bind to specific motors. So there's a specificity there. Intermediate filaments organize into long, several micrometer, bendable, and highly stretchable bundles. So again, you get bundles with a highly nonlinear force stretch behavior. So that means that when you stretch them, the output is not linear. It's, it can be nonlinear. You can have really high output of force or of power from the force stretch, and extensions of up to 3 to 4.5 fold. So there's a pretty large range there. The IF, this specific type of microtubule intermediate filaments turn over much slower than the normal cytoskeletal elements, in the order of hours, and unlike other cytoskeletal filaments, they lack stable and specific linkers. And then they talk about some stretching experience in epithelial monolayers. This suggests synergistic interactions between the actin cytoskeleton, controlling epithelial mechanics at moderate stretches, and these IFs, providing load-bearing and very large cell deformations. So they actually might likely play a role in tensegrity, these intermediate filaments. Under extreme cellular stretches and over long times, the IF network rearranges itself into a star-shaped structure where thick bundles radiate from a central tight tangle. Laser ablation shows that such IF bundles provide structural integrity, so if they ablate them, they fall apart to superstretched cells as the actin network becomes progressively diluted. So this is where you get most of the actin network falling apart. You can still have IF bundles serving to preserve the structure. So they talked about the mechanics of this IF network alone. They go through some of the what they're looking at here. They're looking at strain and stress, and then stress strain curve. And I wonder if they have one here. Well, they have these. This is the influence of filament entanglement on cellular mechanical response. So here we have these different networks, and we have their topology here, where they're stretching. They have this intermediate role, and then you have for the green, the blue, the black, and the red, you have these different functions that show TC versus EC. So EC is cell areal strain, and TC is cellular tension. So areal strain, as that increases, cellular tension goes up, and it goes up faster in this purple version. The red version here, which is here, goes up faster than, say, like the green version, which is here. So you get this more diverse connectivity, and it's, I guess, less, the normalized cellular tension falls off. When you have these sparser networks with bigger bundles, you get more cellular tension. I guess that's the way you interpret that graph. But anyways, they tested a number of different topologies and show them here.
Oh, I can't see the paper.
Yeah, I'll send it to you after the meeting. Or do you want to see the title?
We usually show the paper on screen, and somehow it's not there.
Oh, maybe I'm not sharing my screen. Okay. There we go. All right, so yeah, there we go. Yeah, so this is the graph here, figure 2. Again, this is the epsilon C versus TC, and these are the different topologies for these networks. You have a case where it's sparser, but you have these thicker bundles, versus something that's more highly densely connected, but smaller connections, no bundles, or very limited bundle size. And so where you have a sparser network of more bundles, you get a higher TC, which is this cellular tension, normalized cellular tension. And this is cell areal strain. So this number, as this number increases, the area increases. And so that's what they're showing here. Any comments on that, Susan?
No, I'm just really glad to see a graph with numbers on it.
Yeah.
Yeah, this is what I put the reference in the chat. that's what they were talking about. When you get to 0.7, that much stretch, then the intermediate filaments basically take over. I guess it's because it's diluting the actin network.
Yeah.
At that stretch. And actin is quite brittle, so if you pull on it too much, it'll break. But I also have a paper that shows that the intermediate filaments kind of interweave themselves with the actin network, like just with the actin fibers. So there's different mechanisms probably where the intermediate filaments come into play.
Yeah.
So it's I don't know, complicated.
Oh yeah.
Yeah.
Yeah. So they're using a specific like experimental setup here where they're doing this ablation study. So it's kind of like, I don't know if you'd find these conditions in nature, but I mean, and just showing they're differentiating between the actin filaments and the intermediate filaments. So and then they work out some measures here: pairwise Gaussian linking per number of filament pairs, and then they show this, and they have some sort of supplemental videos that they show this in, and so they're actually looking for network deformations and things that are independent of network deformation. So this measure is actually independent of network deformation. So you can deform in different ways and still get the same reasonable results. This is the mechanical response again, where they're looking at the same measure, mean plus or minus standard deviation of 8 model realizations. They're looking at a 1D model and filament strain distribution. So this is, so this is when varying. So the different things that they're varying here, these numbers up here, LO for different, so they have 4, 5, and 6. And so they're kind of testing these different strain distributions and they're getting different results. So basically, in this case, strain versus, or different types of strain, you get this areal size, and then you have the TC parameter, which increases for 4/6. So as this number increases, as this number decreases, this TC goes up. So you see that for N sub F, which is this, I guess, the filament strain force relation. And then for this number here, LO, which is the distribution of filament lengths. So you have, where they want to look at, the filament lengths, the distribution of them because they're not all the same length, and then the strain force relation. So that's that graph. And then this shows network reorganization for different shapes of the enclosing cell. This corresponds to regular tessellations of the plane. So this is where we kind of get towards the tensegrity aspect of it. So you're looking at these different shaped cells, or these. So this is the enclosing cell. This is a triangle. So NCB, which is the sort of the number of vertices that you have in this enclosing cell. If you have this triangle, you end up with this kind of a network of intermediate filaments, and it looks roughly optimal to this triangular shape, and they're bundled together in a very sparse network. But if you see that when the network changes to a square with 4 nodes, 4 corners, you end up with a different type of network organization. It's still sparse and in bundles, but they kind of attach in different ways. And then you have this hexagon, which is 6 points or 6 corners, and you get this different type of network topology, which is kind of a mix of the two, where you get this cross, and then you get this straight line across, and they're not central to the sides. So you can see on the hexagon, they're organized in a way that's not quite central to the middle of each edge, but they're still, this is like the optimal network reorganization pattern. So that's what they show there. Their work suggests through entanglement and self-organization, IF networks provide a safety net for cells against extreme strains. So this is when the actin networks fall apart and you get this kind of safety network. Being rooted in network topology, this emergent material property is complementary to the role of IF's safety belt against fast strain rates, which hinge on rate-dependent mechanics of filaments and bundles. And so in beyond biology, people have looked at network entanglement, and this has been leveraged to enhance the mechanical properties of hydrogels, and is at the core of textiles. So there are some different innovations in textiles where they're stronger and more flexible, and those are based on this principle. So they identify what they call corralled entanglement, which is something like this, as a scale-free principle for extremely deformable bio-inspired materials. So this is a scale-free principle in network theory that just means that you don't have to have a characteristic number of nodes or links. It's basically has the same type of organization no matter how much it scales up or down. And so this is where we get this this sort of the power of this method and understanding what we're doing here. So, and this, we can think about this as something that is applicable to weaving and knitting at a molecular scale. So in nanotechnology, there's we want to weave or knit different materials together, different fibers, and we want to figure out the most optimal way to do that. So any comments or questions about that? Okay. So then the other paper in this folder is, this is, I don't know if I'm going to be able to get through this paper, but I'm going to just kind of give a very high level description of what it is. So this is integrator-mediated clustering of poised RNA polymerase II, which is the Pol-II, which is the thing that's involved in transcription, synchronizes histone transcription. So this is from what they call systems genetics, and we'll read through it, and I'll give a very high-level overview, but this will be kind of an interesting paper because it had some interesting graphics associated with it, so hopefully we'll be able to see some of those. The abstract reads dynamic clustering of transcription players, including mRNA polymerase II, which is this Pol-II. And again, it sits on top of the unzipped DNA helix and helps to transcribe the genes or whatever is being transcribed. And so this is a molecule that exists in the nucleus. This is thought to rely on multivalent interactions of their intrinsically disordered regions, thereby enhancing active transcription. So this is where you get this dynamic clustering. It allows for sort of this interactions in intrinsically disordered regions, and this enhances active transcription. So this is a mechanism to enhance transcription through some, there being more Pol-IIs in a certain place. Using the histone locus bodies, HLBs, of Drosophila nurse cells. So they're looking at Drosophila nurse cells here. It's a certain type of cell in Drosophila, and they're looking at histone locus bodies. So, this is, histones, of course, are involved in epigenetic regulation. So they're using that kind of model. We find that Pol-II forms long-lived transcriptionally poised clusters, distinct from liquid droplets. So they compare this with liquid droplets. We've talked about liquid droplets in the past and some of the things that they serve in biology and cell biology, which contained unbound and paused Pol-II. So these different clusters, they have a number of Pol-II. Some of them are unbound, some of them are paused. Depletion of the integrator complex endonuclease module, but not its phosphatase module, are pausing factors disperses these Pol-II clusters, leaving HLBs intact. So this is where we have this, these clusters get dispersed, but the HLBs, the histone locus bodies, are intact. Consequently, histone transcription fails to reach peak levels during S phase of cell division. So this is where your histone transcription fails when it's dispersed and aberrantly spills over throughout the cell cycle. So this is where these clusters are dispersed. There's a lack of peak level of histone transcription during the S phase, but then in other parts of the cell cycle, it spills over and it's, I guess, enhanced. I don't know, but basically the cell cycle is disrupted, or regulation of the cell cycle is disrupted. We propose that clustering sequesters numerous poised Pol-II molecules near gene promoters to ensure synchronous and efficient gene activation at desired times. So, there are a lot of regulators of eukaryotic transcription. There's one of those is Pol-II. There are other co-activators and other sequence-specific transcription factors, and they can assemble into these membraneless clusters, and they can be compared to water droplets. These clusters are mostly highly dynamic, short-lived, and exhibit liquid-like properties, so they come together, they don't last very long, and they shift around a lot in terms of their size. Clustering of transcription regulators is dependent upon the multivalent interactions of their intrinsically disordered regions. So they have these disordered regions, and is generally thought to be mediated by a liquid-liquid phase separation. Since its initial observation, clustering has been thought to facilitate transcription by allowing for rapid accumulation of high concentrations of these Pol-II molecules at the target genes. So you need something to transcribe things. If they're near the target genes, you have more of them to transcribe and do the work of expressing the gene. This has been supported by recent observations that artificially induced clustering of transcription factors is sufficient to boost transcription. So you can actually use like an artificial clustering technique where you cluster transcription factors and you can boost transcription that way. So this has been done artificially, but they're using this nurse cell model to do this. So they talk about the biochemistry here. But basically what they did in this paper was they leveraged microscopy with genetic perturbations in the Drosophila ovarian nurse cell to investigate the dynamics, assembly, and activation of Pol-II clustering at the histone locus and its regulatory roles in transcription. So I think we had something in the chat. Dick says these various arrangements of IFs, and this is back to the last paper, have consequences for, and then he cited this Rutland and Gordon 1990, intermediate filaments may prevent buckling of compressively loaded microtubules. Comments. So yeah, I don't know, it'd probably be a Susan question. This is like a paper from 1990, intermediate filaments may prevent buckling of compressively loaded microtubules. So this is again these intermediate filaments, and we had this. Okay, thank you for attending. We had this mention of that it's like the safety factor, and so this is something that we might have more conversation about later. I don't know if you want to think about that, Susan, or if that's I'll think about it as well. I don't know right now.
It looks like it ties in for sure. I don't know whether I should put it in my candidacy exam. They'll just ask me a whole pile of questions about it.
Yeah.
That's the problem. But I'll put that in my notes.
Okay.
Yeah. The relevant paper, and I have to do some more on this topic. So more about that later, I guess.
Yeah.
But the first, the one you presented with the knitting and how things are entangled certainly fits in. And of course, Yeah, I don't know, just it fits in with something else I was reading about tensegrity structures,
Right.
which
Yeah.
can be like a bird's nest, and yeah, Kagome structures, Kagome networks. Yeah,
Kongomi?
Kome.
Komagomi,
Yeah, okay. I'm not
maybe.
very good at this. I'll find it for you. Let's see. Yeah, it's hard for me to go ahead and continue. I'll
Okay.
try to find this.
All right. All right. So yeah, let's go back to the liquid droplets paper, just to kind of finish up on it. And so yeah, so basically what happens is that these clusters, play a role in transcription, and they're involved, of course, differentially in different cell cycles. So in ovarian nurse cells, these are in developing egg chambers in Drosophila. They provide a unique natural system for capturing Pol-II clustering without the need for either of the things that they're yeah. So the NCs are endocycling cells with alternating G and S cycles. So this is where you have a cell cycle of 2 cycles, G and S. And then this is where they can grow to 1,000 N ploidy, which means that they have this a lot of gene duplication and reach 50 microns or more nuclear diameter. So it's a pretty big nucleus. And of course they need it for this sort of ploidy that's going on in the cells. Oops. And so this is something. So the most prominent Pol-II clusters often reside on the replication-dependent histone genes. So again, this is a model cell where you have a lot of ploidy. You have this huge nucleus, and these clusters are in the nucleus, and they're involved in sort of transcribing. And so the most prominent Pol-II clusters often reside on the replication-dependent histone genes, which in Drosophila are present in around 100 copies at a single locus. So this is something that needs to be transcribed a lot in each nurse cell. The histone locus self-organizes into membraneless subnuclear compartments called histone locus bodies, or HLBs. And so these are these clusters which form one visible focus per NC, or nurse cell, in early egg chambers, and multiple foci per nurse cell in mid to late stage egg chambers. So you have these one focus per nurse cell. In early egg chambers, you get multiple clusters per nurse cell as we progress in development, as DNA copy number increases. So as that copy number starts to increase, the ploidy starts to increase, then you get this, you get more of these clusters. And so basically it's sort of building a storehouse of Pol-II so it can transcribe all these loci that are being produced, and this is playing a role in development. The repetitiveness of Drosophila histone loci combined with the polyploidy of nurse cells thus constitute a naturally occurring signal amplification platform for imaging these clusters. So this makes it really accessible, and they can observe this at a very it's almost like high throughput biology, but just with the biology being the high throughput part. So they do some fly genetics. They create a specific line that's allows them to do these experiments. And they mention that nearly all the HLB-bound Pol-II molecules are transcriptionally inactive. So the observation that Pol-II clusters are present at the HLBs, or the histone locus bodies, and the majority of G-phase nurse cells suggests that a fraction of the Pol-II clusters are transcriptionally inactive. To confirm this, we measured SIR2 phosphorylation levels at the HLBs, And this marks Pol-II molecules that have advanced beyond the promoter proximal regions of genes. Amongst all nurse cells analyzed, only 2.9% show visible levels of enrichment over nucleoplasm, suggesting that high levels of histone transcription only take place during a tiny fraction of the clustering lifetime. So these clusters are there for maybe like, maybe it's a probabilistic thing where you need to have a lot of them, but only a small fraction of them are, transcriptionally active, or maybe that, you need to have the structural integrity to have, to transport these things to the proper locations, or maybe that they might serve other functions in other cases. But basically, you have these clusters, a small number of them are active, but then they're able to do, so this is why when you get more and more copies of the gene, or more copies of the locus, you end up with more clusters. And so this is where you're getting an increase in Pol-II in the clusters, along with an increase in the genes. So these data argue that Pol-II clustering occurs before pause release, with most Pol-II molecules remaining inactive for a substantial period following recruitment to the histone locus bodies. So this is where they kind of lay this out. And I'm looking maybe for some images here. They don't really have figures in here. That's probably down further. Okay, so this is a nurse cell here. This is what they look like. You have sort of stage 2 to 4, stage 5 to 10, and stage 11 through 14. So the nurse cells are these white cells that are, kind of grow in size, and then they're depleted by stage 11 and 14. And so this is where they're imaging the nurse cells. They're imaging the, this is where you have the Pol-II cluster. And so you can see in the G phase and the S phase, the Pol-II cluster is present throughout the S phase, but only part of the end of the G phase and part of the beginning of the next G phase. So it's not present throughout all the G phase, but all throughout the S phase. And so this shows some of the these clusters. This shows the intensity of these different variants, the histone locus bodies relative to wild type Pol-II, normalized by expression levels. So you see that you have the clusters, you have these different Pol-II variants within the histone locus body. And then this just shows kind of more of the same. You see the expression here in these bodies. All right. So that's all for that paper. I just thought it might kind of tie into some of the structural biology we've been talking about. And I thought it was kind of interesting. But it's a different system from the first paper. So.
Okay. It's Kagome structure.
Okay.
It's a weave, type of weave.
Yeah, Kagome structure. Okay.
Yeah. It has interesting properties.
Yeah. Yeah, that might be interesting with specific application to tensegrity. But yeah, it mentioned about it being relevant to weaving and things like at the nanoscale, things like that, microscale.
Yeah, you can make metamaterials with some of these structures.
Yeah.
It's really strong artificial materials.
Yeah. Okay. Are there any other comments or questions before we go for today? So now I'd like to talk about a paper that's related to something Susan said in the meeting. She was talking about metamaterials and how we can use some of those intermediate filaments to weave them together into very strong materials. And so that's metamaterials, and that's an emerging area of nanotechnology, microtechnology. But this is an interesting paper on metasurfaces, and specifically adding the sort of the dimension of morphogenesis to the creation of metasurfaces. And this paper, Nature Communications, this is from this year, Morphogenetic Metasurfaces, Unlocking the Potential of Turing Patterns. So we revisit the reaction diffusion principles of Turing patterns to talk about creating these metasurfaces, patterning surfaces in different ways that leverage this morphogenetic aspect. So let's zoom in on the abstract. So the abstract reads, the reaction diffusion principle imagined by Alan Turing in an attempt to explain the structuring of living organisms is leveraged in this work for the procedural synthesis of radiating metasurfaces. So procedural synthesis is basically like a generative model. And so they use procedural synthesis in art a lot, in AI art and other types of three-dimensional modeling. And so what they're going to do is build this radiating metasurface. So it has like a pattern on it that changes over space. The adaptation of this morphogenesis technique ensures the growth of anisotropic cellular patterns, which means that they're not the same everywhere in the pattern. Automatically arranged to satisfy local electromagnetic constraints, facilitating the radiation of waves controlled in frequency, space, and polarization. So this is where, we want to have this anisotropic pattern. We want to make sure that for reasons that you might use the surface as a substrate or, nanoelectronics or other types of applications like that, we want to have something that conforms to local electromagnetic constraints. We also want to be able to, have a controlled sort of pattern that is useful for different types of things. So experimental validations of this method are presented, designing morphogenetic metasurfaces, radiating far-field circularity polarized beams, and generating a polarization multiplexed hologram in the radiative near-field zone. The exploitation of morphogenesis-inspired models proves particularly well suited for solving generative design problems. So again, this is where we want to build something. We don't exactly know what the actual design will look like, but we can generate a number of potential solutions and come up with an answer. And of course, we use generative techniques for that, but this is one such technique. We also want to convert global physical constraints into local interactions of simulated chemical reactants, ensuring the emergence of self-organizing meta-atoms. So, of course, we can use reaction diffusion as it was sort of intended as a chemical design mechanism or as a chemical self-organizing principle. So, now, of course, Alan Turing created this mathematical framework. It's been proven effective at reproducing the spontaneous formation of numerous biological patterns. So we know from previous meetings that there are these Turing patterns that, kind of come out of random assortment of molecules, and they're exposed to these morphogens, these antagonistic chemical species that react and form gradients, and those gradients serve to organize the array in different ways. So depending on the concentration of these morphogens, which are these theoretical constructs, so they can be anything you want them to be. They can be some sort of specific signaling molecule. They can be a protein, whatever. Depending on their concentration, they interact to form diverse patterns. And there's this aspect of predator-prey dynamics in there, which is basically if you have two different morphogens and they can form, say, a gradient, or they can compete for each other. They can compete to out-compete each other and form different patterns that way. So depending on the concentration, if, say, one is bound by the other, and then one is overproduces compared to another, then you get this sort of predator-prey dynamic. And I don't know what the citation is here, number 5. We'll look later and see what that is. The effectiveness of such models for the synthesis of biological characteristics and functions relies on the decentralization of the instructions guiding the structuring of life. So we don't really have a centralized control mechanism. This is all very decentralized. So this is the point that they're making here. Dussart et al. Have conceptualized the development of such morphogenesis-inspired generation techniques based on self-organizing structures to simplify the engineering of complex systems, and those are references 7 through 9, which we'll also come back to. They talk about this. They talk, of course, about not only the Turing model, but the Gray-Scott model, which, of course, is another variation of the Turing model we've talked about in previous meetings. And so this is where we get a great variety of reaction diffusion systems that can be generated. And so people have worked on classifying the patterns that come out of the Gray-Scott model. So we have a guide for sort of this pattern classification, different types of reaction diffusion leading to different types of patterns. And so, this is something that we want to be able to adapt this model to specific constraints. So this is why we're interested in classifying these patterns. In this case, it's in terms of some sort of electromagnetic constraints that we have to satisfy. So this is an example here, figure 1. This is constrained generation of anisotropic Turing patterns. So this is the Gray-Scott model, and this is simulating these morphogens, or these antagonistic chemical compounds. So you're just simulating them in different ways. You're simulating the concentration, their action. So you have this set of reactions. You have this generation of particles. You have an extinction rate, which is K. So this is much like in the Volterra model, the predator-prey model. K is usually deaths. This is extinction. So we also have generation, F. So in the predator-prey model, this is births. And so we have this birth process and this death process that are competing. So you have an extinction of one type of molecule. You have the birth of another type of molecule. The blue molecule will say, well, it will compete the orange molecule. So this is the way they set this up. And then there's diffusion, which is where you have diffusion that's differential. So the blue particles they diffuse more than the orange molecules. And so they outcompete them in terms of diffusion. They're in more places, and you can control that parameter to get this type of anisotropic generation, which you see on the right, where you get this pattern that's asymmetrical and anisotropic. And so you can see here that you have one side with the orange, you have one side with the blue, and that intersects in different ways. And of course, for Turing patterns, we're able to form the script for Turing within the pattern, and this just shows that you can finally control how these things are expressed. So they're doing procedural generation, and they're trying to guide the growth of these patterns. They're going to etch this onto a very thin, small plate, and they're arranging this between 2 thin copper sheets, so they're able to get this sculpted. And this is an example here in figure 2. This is morphogenetic metasurface characterization and synthesis. So in step 1, we have a diffusion tensor, which defines our morphogenetic parameters. We have pattern type, which is controlled by F and K, which is controlled by the growth and death of the molecules. We have pattern size, du and dv, so we have control over diffusion. We have pattern anisotropy, which is the relative asymmetry of the image of the pattern and how the pattern changes across space. And then we have an orientation parameter, theta, which is this, or phi, which is this idea of like how it's oriented in space. So step 2, of course, we have periodic pattern growth, which is oriented in a certain way. It has a pattern size, pattern type, and pattern anisotropy. Then we do a finite element simulation, which is step 3, which is where we have this, where we break the pattern down into specific elements, and we model the mechanics. And then step 4 is a characterization of associated reactance tensor. So this gives us our electromagnetic parameters. So from the FEM simulation, we do the physical modeling. We also can do some electromagnetic modeling, and that leads us to step 4, where we define our average reactance, which is X sub SW, reactance, anisotropy, and orientation, once again. And then in this part here, B, is where we take this objective surface reactance, which is this average reactance. We have this feed mode, and then we have this objective electric field, so we test it out, our different designs. And then we have this metasurface generation, where things are actually, there's a deposition on the surface of these different patterns. And so this is where, this is down here, they talk about definition of radiation objectives enable the desired electromagnetic characteristics to be derived at the surface of a parallel plate waveguide excited by a monopole. So this is what they're doing in the synthesizing the thing onto a surface. So they're basically depositing this thing by a monopole system. Conversion of these targets into local morphogenetic parameters are required for metasurface generation. So you need to have a target but onto a local set of morphogenetic parameters. Iterative resolution of the anisotropic Gray-Scott model ensures the growth of elliptical patterns, which you can see here, undergoing a series of successive divisions until they occupy the entire permitted space. So you start with a pattern and then you replicate that pattern through successive divisions. Under the influence of the morphogenetic parameters, the Turing parameters self-structure to synthesize the desired electromagnetic tensors. And so this is an example here in this figure. This is a demonstration of procedural generation of morphogenetic metasurfaces. So this is metasurface. A is a metasurface radiating a broadside LHCP beam, which is here. So this is radiating this beam. This is where they do the hologram part. Dual polarized metasurface radiating LHCP and RHCP beams simultaneously in 2 different directions. So this is where you get a beam that you're shining on this pattern. In this case, it's in one direction. In the other case, it's in 2 directions, as you can see. And this is the sort of the inset of this area. So at the top, you have this part of this pattern. We have the same part of the pattern, but radiated by 2 different light sources. So you can see the first light source, and then the second part where you have 2 light sources. And then 3, you have a color map representing orientation and anisotropy of objective tensors. And this just shows the direction of the principal eigenvector versus anisotropy over that surface. Okay. So they give their methods. This just shows a demonstration of a polarization multiplexed hologram at 20.2 gigahertz. This is where they're shining this light beam on there and creating the hologram. And they're actually able to create shapes on the surface. So you have an L and an R. And so you can see that they're able to do this. We have this measured electric field distribution represented according to a left-hand circular polarization, which is here, and a right-hand circular polarization, which is here. So they try to do different polarizations to make this work. And so they have their methods here. This is where they talk about how to develop and validate this work. And definition for objective reactance tensors. This anisotropic pattern synthesis. So they use the Gray-Scott model here, and they define this. Then they're able to look at diffusion, which is by defining each point of the space with an associated tensor D, decomposing the Laplacian operator into 2 successive gradients, shown here. And then there's a direct correspondence between the eigenvectors of the diffusion tensor and those of the generated reactance tensors, allowing the following diagonalization of the matrix. And then this is where you get your eigenvalues, and then you can do some of this other stuff. Characterization of the anisotropic patterns. So this is where we have the average reactance and the anisotropy modulation coefficient. And these are stated in this way. There are these different reactances, and they're taking the average. In this case, they're taking the absolute value of S1 or X1 and XSW, which is the surface wave reactance, and then averaging that by the surface wave reactance. So AX is this anisotropy modulation coefficient. And so then we have these other measurement techniques that they use, and they have data available for this, and they also have code available. And so if we get onto our references, we see that they have some nice references. Kondo and Miura, which is a paper on using the reaction diffusion model to understand biological pattern formation. Of course, they have the Turing paper, Chemical Basis of Morphogenesis. This is a 1990 version, which I assume is a reprint of the original. We also have Hough genes regulating digit patterning by controlling the wavelength of a Turing-type mechanism. So people have done this with different types of methods before. And then we talked about 6, which is colonate and liquid, which is programmed in self-organized flow of information during morphogenesis. So this may be a very interesting paper to look into for future meetings. René Drosophila has some interesting things on programmable architectures that are complex and self-organized from morphogenesis to engineering. So this is an A-Life paper from 2008. Drosophila, Hiroki Sayama, and Michael, a review of morphogenetic engineering. This is from Nature Computation. This might be another interesting paper. And then this paper by another set of co-authors, Programming the Emergence in Morphogenetically Architectured Complex Systems. So we have a number of papers where they kind of work these methods out. And this is kind of following up on that to create these metasurfaces. We also have some interesting resources on agent-based modeling of morphogenetic systems. This is a 2019 paper. Gigavoxel computational morphogenesis for structural design. So this kind of focuses on the design aspect. There's also this paper on the one-dimensional Gray-Scott model. So this is pattern formation in the one-dimensional Gray-Scott model. That was in nonlinearity in 1997. So this goes back a while. And then we have reaction diffusion textures and other types of reaction diffusion textures in computer graphics, where they've used reaction diffusion as a way to create two-dimensional textures and three-dimensional textures. Okay.
Oh well,
yeah.
I'll say bye, and yeah, I have to finish writing up my my paper.
Okay. Good luck.
Thank you.
All right. Thanks for attending. See you next week.
Bye bye.
Bye.
Bye.